low coverage shotgun sequences, and those are what we use to design our nuclear loci for the phylogeny that we've previously presented. But we actually do have a whole genome of Penstemon now, and it's been kind of a fun adventure because I'm kind of late to the game of genomics, and I really didn't know what I was getting myself into, to tell you the truth. But let me start with the really cool things about Penstemon. I've presented work on Penstemon for my entire career, but I just want to emphasize once again, it's the coolest genus in North America. It is actually, despite what the... I have implicit bias here. Despite what the new treatment for flora of North America states, that's North America north of the Mexican border, Penstemon truly is at this point the largest genus that's native to North America, occurring from Alaska down to the southern highlands of Mexico as it borders with Guatemala, goes coast to coast, exclusive of the Canadian Shield because it got scraped off into the bare earth when the glaciers were there. But if you kind of look right here, that's the epicenter of diversity, the Intermountain West. And unfortunately, I'm over here in Ohio with only 6 species. So it's really, truly intriguing because it's a very young genus. We've dated the origin of this genus to about 2 million years ago, so somewhere between the end of the Pliocene and the beginning of the Pleistocene, and we have nearly 300 species. So I just want to give you some pictorial porn here of how beautiful Penstemon is. It has a wide diversity of morphologies, from habit to leaf morphologies, to inflorescence morphologies, to, get this to switch, there we go. Isn't that beautiful? That is the coolest genus, I swear to God, I'm so lucky to work on this genus. And a lot of the diversification we've hypothesized has been due to pollinator selection and the pressures on floral diversity. But if you were in the session this morning on macro evolution, my student Ben Stone is actually working on adaptation for some of the other characteristics. And it turns out, sorry, I'm flashing somebody, that there are other factors at work besides floral evolution. It has a wide array of staminodes, 300 species, and a lot of differences in the staminode. The common name for Penstemon is beard tongue. The staminode actually kind of sticks out of the corolla for some of the species. It's kind of hairy, therefore the beard tongue. But not all of the staminodes have trichomes. Some of them are actually glabrous, and some of them are very short. So there's a lot of variation on that morphology as well. I've presented this tree before, and our phylogenetic analysis, we have now in our data set almost 96% of the genus. So we really have very good coverage for the phylogenetics. And if you'll just kind of take a look at this part of the slide, this is our tree. And so it's really, branch lengths are very short. This is based on 42 nuclear loci. And some of this area here is really difficult to resolve just because it's such a recent radiation. Traditionally, the genus has been divided into 6 subgenera. In 2006, I published a phylogeny where I suggested that maybe we're looking at 2 subgenera, and in the Flora of North America treatment, that's what Craig Freeman has recognized as 2 subgenera. So this isn't about phylogenetics. Basically, we're trying to go to the next level, the next step, and so we wanted to get a genome sequence for Penstemon as a starting point for further studies. And so basically, we wanted to find a genome size that was relatively small and where we could actually get material to sequence. And so if we look at the diploid genome size, it ranges from about 462 megabase pairs to about 919. And then we have a whole lot of different polyploid complexes in the genus with genome sizes from 1148 to 3152 megabase pairs. So obviously, Penstemon digitalis, which is a dodecaploid, is not a candidate for our first genome sequence. So we selected Penstemon petiolatus, which has a genome size that was estimated from flow cytometry at about 719 megabase pairs. Now, we didn't select this from an evolutionary standpoint. We selected this for convenience sake because we were starting this project in the middle of the winter or at the end of the outside of the flowering season. There was no way for me to go in the field to actually collect tissue. So I went to Brigham Young University to colleagues of mine, greenhouse, he happened to have a whole bunch of different species in cultivation. The tissue for Penstemon petiolatus was prime. I'm not sorry about that because look at this species. Does that deserve a genome sequence or what? I mean, it's absolutely incredibly beautiful. But it's actually placed here toward the more derived end of the tree. So down the road, we will go deeper into the evolutionary history. So basically, collected a lot of fresh tissue. I mean, I think I collected 10 grams of tissue on liquid nitrogen, flash froze it, sent it to Dovetail Genomics, who did the high molecular weight DNA extractions, and then they did all of the sequencing, PacBio, Chicago, Falcon, all those names that I have really no clue about what they mean, except that the product that we get back is really good. And so basically what they gave us was a library or an assembly that was incredible. So they gave us 619 base pairs back of that 719 estimate. That's a lot of data that came back. We used the Maker pipeline. I'm thinking I missed a slide in here. Nope, I didn't. Actually, I sent all the data to Paul Blischak in Mike Barker's lab. He was my grad student, now he's a postdoc at Mike's lab. So he did all the data analysis, and he's not here to actually answer any of your questions because he went over to Europe to meet his fiance for a vacation. So anyway, he did most of this work with the pipelines and annotations. So we used Maker 2, did several iterations for gene prediction, and then ran it into Maker 2 again to generate the annotations. And then MC-Scan was used to look at the intragenomic comparisons and intergenomic comparisons with Antirrhinum majus. Lucky for us, the whole genome sequence for Antirrhinum was published earlier this year, and it was a really nice genome. So it's in the same family, Plantaginaceae, not too distantly related, and so we could actually use that for comparison. So we had annotated the genes from the 8 largest scaffolds of Penstemon and compared that to the annotated genes on the 8 chromosomes of Antirrhinum majus. Now, Penstemon also has 8 chromosomes, so we were really hopeful that we would have kind of a linear progression. The final assembly for us was about 615 mega base pairs into 1,934 scaffolds. The N50 value was 71.3 mega base pairs. That's pretty good. And our average coverage was about 30x. Our longest scaffold is probably a single chromosome, 103.3 mega base pairs. And all of the 8 largest scaffolds were much larger than 10 mega base pairs. So what we have here are 8 contigs. Not all of them are completely assembled into chromosomes. At least one of them probably is. So we have not perfect assembly, but we have pretty good chunks and a pretty good result here. So using Maker, we assembled 25,892 putative genes. Compared that to about 37 or 38,000 for Antirrhinum. And this number is probably going to improve when we're at the end of the study. We're just at the beginning of these annotations, so there's a lot more work to do. So I'm going to just kind of do some of the comparisons here between the intragenomic comparison with Penstemon petiolatus to itself, and then Penstemon petiolatus to Antirrhinum majus. Now the reason we want to look at it compared to itself is we want to see if there have been duplications in the genome, and so this is one way to do it. So the first thing you do is a Intoning plot, and we have the 8 largest contigs mapped against itself, and there should be a line here going down the center. So we're just going to look at the top half of that graph, and so if we look at contig 1 right here and map it to itself, you can see that there are some chunks that are actually in different places on that contig that have been duplicated. And you map it across, I'm having a hard time with this. You map it across these different contigs, and you see there's big chunks, and that's the way it is all the way through that upper right-hand corner. It's a mirror image on the lower left. This is another way to visualize that. All these lines are representing duplications, either within the same contig or among other contigs. So for the intragenomic comparisons, we show large blocks of genes that are retained in duplicate across the genome, and that indicates that there's some chromosome rearrangement and fractionation probably after a whole genome duplication. This is the comparison for petiolatus with Antirrhinum majus, and we have the 8 chromosomes of Antirrhinum here on this axis, and then the largest contigs for Penstemon along the x-axis. And you can see that there are big chunks that are in common, but they are in different locations from that 8 chromosome arrangement. And here's what that comparison looks like. So there's a lot of mess here. And this was a little bit surprising because we were hoping with a close relationship that we would actually be able to do a little bit better job of comparing in a linear fashion. But it's certainly not that easy to interpret. So we did a comparison of the syntenic blocks between those 2 genomes. And it turns out for Penstemon compared to Antirrhinum, there's 2 duplications or 2 copies of loci, and when you look at Antirrhinum, there's 3 compared to the Penstemon. A little bit confusing, but the ratio of syntenic blocks between these genomes give us a little bit of evidence for maybe more than 1 whole genome duplication. So I don't really have conclusions. We're still in the preliminary phases of our analysis pipeline. Maybe I'll have a much more complete picture to report to you next year. But I want to just share some thoughts about where we are at this point. So it seems like we've captured most of the Penstemon petiolatus genome. We probably have missed a lot of areas of repetitive DNAs. Probably we're having trouble with the assembly in the centromeres or the telomeric regions. There is evidence of ancient whole genome duplication in Penstemon petiolatus. And we really do need more genomes for Penstemon. It'd be nice to have more genomes across Plantaginaceae as well. So we're still in the early stages of analysis, so we need to complete our annotations, we need to do the blast searches and identify the gene classes, but those results will actually be very useful because one of the things we're interested in doing is looking at adaptation and trying to identify genes that might be associated with particular morphological traits or physiological characteristics. The second page of thoughts here is that the 1KP study actually showed whole genome duplication that preceded the formation of the family Plantaginaceae. And Lee et al. In their sequence for Antirrhinum that was published in Nature Plants inferred a Plantaginaceae specific whole genome duplication, but their sample size was 1, that was the Antirrhinum genome. So it's possible that our results are supportive of this finding, but definitely more analysis and taxon sampling is going to be needed. So with that, I'd like to acknowledge our funding sources. Members of the Wolfe Pack have been really instrumental in helping me through all of this. My collaborator, Laura Kabako. And then Mike Stevens provided the tissue for us and has been very helpful in the field. And then the Barker Lab has also been instrumental in helping us with the annotations. So thank you very much, and I'm sorry that I have to run out.